This is Jonathan Already. This is my Bio-5 lab project. For this experiment, we will be experimenting with osmosis. Osmosis is a form of cell transport, more specifically a passive transport. Passive transport occurs when substances cross the plasma membrane without any input of energy from the cell. No energy is needed because the substances are moving from an area of higher concentration to an area of lower concentration. Osmosis is a specific type of simple diffusion categorized under passive transport. It is the passage of water from a region of high water concentration through a semi-permeable membrane to a region of low water concentration. Water moves in or out of a cell until its concentration is the same on both sides of the plasma membrane. Semi-permeable membranes are very thin layers of material that allow some things to pass through them but prevent other things from passing through, such as oxygen, water, and carbon dioxide are capable of passing through, but they do not allow larger molecules like glucose, saccharose, proteins, and starch to enter the cell directly. So for this experiment, the example we will be using is a sweet potato and placing it in 3 different solutions to see the reaction. A solution is described as a mixture of water and a solute. The first solution will be a sugar solution, the second solution will be a salt solution, and lastly we will have just tap water to use it as a control. The materials being used for this experiment will be 1, 2, 3. I know it's hard to see because they're clear, but there's 3 of them. We're using 3 cups. We're using some salt, some sugar. So the salt will be for our salt solution, sugar will be for our sugar solution. Got tablespoon for measuring the solutes, and then I don't know if this is needed, but a chopstick, and that's how I'm going to use it to mix the water and the solute for the solution. So first we'll start by making the solutions, right? So I went ahead and off camera filled up our 3 cups halfway with water. Secondly, we will grab half a tablespoon of sugar solute, place that in one of them, and then we will grab half a tablespoon, measure out half a tablespoon of our salt solute for our salt solution, and place that in a separate cup. So now we have our salt solution that will be mixed very shortly. We have our sugar solution that will be mixed very shortly. And lastly, we have our water or control. So we'll do the mixing and we'll get into placing these sliced potatoes into each one of our solutions very shortly. And then we will wait 24 hours to see the results of the experiment. So it has been about 24 hours. So we're going to take a look at each slice of our sweet potato and the 3 different solutions. First, we'll start with our sugar solution. And right off the bat, I notice it is a lot larger than it was when it was first placed in there. It's very crisp, but so for this potato, the reason it grew in size is because it was placed in a hypotonic solution, meaning the potato was placed in a solution with lower solute concentration, causing water molecules to move into the cell, or potato in this case, which caused it to grow in size and the cell to expand. Right? And so second, let me grab my handy dandy chopsticks, my favorite grabbers in the world. And let's look at our control here, which is our water. And you could see in thickness, it is a lot smaller than the sugar one here. You can see that they were each sliced the same size. And the reason this one did not grow as much in size in comparison to the sugar solution is because it was placed in an isotonic solution, meaning that the potato was placed in a solution that had very similar solute concentration as the potato, ultimately causing no net movement of water molecules in or out of the cell membrane or potato. Lastly, let's look at our salty solution potato. Come here. Okay, I got it. It's a lot thinner in size. It looks like it has shrunk a lot in comparison to the other 3. Yeah, it is a lot smaller. It's very flimsy. So our last potato was placed in the salt solution, As you can see, the potato has shrunk in size. This is because the potato was placed in a hypertonic solution, a solution that has a higher solute concentration than the potato, causing water molecules to move out of the cell, or the potato in this case, causing crenation, which results in the cell or potato shrinking in size. And so that is the end of our experiment here.